Dr.
to hear
Koye,
from you.
we salute you.
Good to hear from you. Cliff, I was asking you how you're feeling. You look good, though.
I'm still alive. And in the Christmas mood.
Yes, that's good. Okay, good to
Thank
see
you.
you again.
Good to see you.
Okay, everyone, I believe we can go ahead and get started with this afternoon's event. So I will begin by, first of all, extending a warm welcome and thank you to everyone who has attended the first of its kind ANPA COVID-19 conference. So again, welcome. My name is Dr. Susan A. Dionwe, and I am the membership committee chair. And I will just take a few minutes to share a very important message on behalf of ANPA. First of all, I would like everybody who is maybe not an ANPA member and those who may be an ANPA member to remember that ANPA has a very rich history of philanthropy and advocacy here in the US and in Nigeria. It's been in existence for over 25 years, has over 20 chapters across the United States. ANPA, by all rights, is an extremely formidable organization. And we're an organization that is committed to evolution and modernization and hearing what our members want. We're committed to providing benefits, quality benefits for our members, such as the programming that we have today and our routine webinars that provide key financial information to our members. So I say this all to say what our ask is. And our ask from those of you who may not be members is we want your support. We want you to join us. We want you to hear what we have to say. For those who are members, we want you to help us to continue to grow. Again, help us to support our growth, not just through your dues, which are very important, but through your voice and through your time. Engage with us on all of our platforms. We have e-groups, we have WhatsApp groups. Most importantly, we have local chapters that will benefit from your time, your commitment, your voice, and your money. So, ANPA really meets a very important need for the Nigerian physician community, the Nigerian health practitioner community, and we need you to believe in us and to be a part of it and to help spread the word. We already know that there is strength in numbers and our collective voices really takes us very far. In this last final moment before I hand over this baton, I do want to remind everyone as the year comes to a close that the beginning of next year is renewal season. So if you have already not put your mind on renewing your membership dues, please do so. You will be getting some regular communications from Letitia, from the membership team, as well as the IT team on new processes for membership payment. From all of that, I say again, welcome. At this time, I would like to proceed by introducing one of the co-moderators for this afternoon's event. He is a board-certified, fellowship-trained orthopedic surgeon. He is a member of the Georgetown MedStar Orthopedic Institute in Washington, D.C. He is the co-founder of the Center for Advanced Specialty Surgery in Nigeria. He is an avid philanthropist. He's the founder of Operation Stand, Walk, Run, as well as the 2019 Humanitarian Award winner provided by HCA. And on top of all of that, he is the chair of the ANPA DMV chapter. He is a rather impressive and formidable ANPA member. I introduce to you Dr. Aham Onyike.
Good afternoon, everybody. Thanks for the very, very flattering introduction, Susan. It is indeed a pleasure to be a participant and a moderator for today's very important discussion on the COVID-19 vaccine. Once again, I'm the chapter chair of Washington, D.C., so I say compliments of the season from the nation's capital. The nation's capital, as we recognize, is the epicenter of COVID. It's the home of the U.S. Congress, which right now, as we speak, is meeting to decide on a $940 billion or possibly up to $1.4 trillion stimulus plan. Here in Washington, D.C., we just had the FDA yesterday approve the Moderna vaccine, which has equal clinical trial record with the currently distributed Pfizer vaccine. Here we have the NIH, which is the forefront of the research and scientific information pertaining to the vaccine. We have speakers from all over the United States that are outside of Washington, D.C., but connected to each other and have emerged in 2020 as the thought leaders, authorities in infectious disease, particularly with COVID-19. And we're proud to have them as fellow Nigerians lead the discussion. So it's an opportunity to piggyback on what Susan just said, that ANPA represents the potential strength in numbers. And it's been on display more so than ever in 2020 with Zoom and other online platforms. And today, I'm very confident that we would all agree after the discussion that we do have the expertise within our ranks. We're going to have a broad and deep conversation on the COVID-19 vaccine, which has been led by many fellow Nigerians, some of whom are here today. In particular, I want to introduce Dr. Getty Anyanwoke, fondly known as Gertie. She is the chair of the, she's the medical director of the Infectious Disease Associates of Baton Rouge, also medical director of the Multi-Specialty Clinical Care Center, also in Baton Rouge. Gertie, in addition to that, is the chapter chair of the ANPA Louisiana chapter, the current committee chair of the Infectious Disease Committee for ANPA, as well as the chair of the COVID-19 task force. Gertie, as you all know, has been very visible, very prominent, very available in leading discussions pertaining to COVID-19 this year, and of which the hottest topic currently is the vaccine. So without further ado, I want to introduce an authority in COVID-19 discussions and the vaccine, Dr. Gertie Anyanwoke. Thank you.
Thank you so much, Aham. That was really flattering. Without further ado, I would launch in to introduce our current Association of Nigerian Physicians in the Americas, ANPA president, no other president than Professor Chris Okunseri. Professor Okunseri is a professor of dentistry at Marquette University. Before we introduce the panelists, we would like to call on Professor Okunseri to give us a brief talk about ANPA. Professor Okunseri, you have the floor, sir.
Thanks, Gertie, and welcome everybody to this great opportunity, and especially thanks to the panelists. But I think another sincere thanks has to go to the Infectious Disease Subcommittee and the COVID task force that Dr. Gertie Anyanwoke chairs. Gertie and that group have been working round the clock from the very minute we identified COVID to now that we have a vaccine. But I also would take this fine opportunity of our great organization to echo all the introductory remarks that have been made so far. Dr. Susan Edianwe reminded us of our great organization and why it's important for us to continue to remain members of this group, talked about our dues, talked about value of being a member of this group. But especially, I think this opportunity showcases all the collective strengths, talents, resources of our great organization. We can see the content. We are going to hear the content from these talents we have and the expertise of the panelists about the vaccine that would help all of us to actually try as much as possible to bring this pandemic under control. So our organization continues to be great. We are very honestly grateful for our membership, without which we would not be where we are today. And I encourage each and every one of us to continue to demonstrate and keep that flag flying. We are all proud of the talents we have in this organization, and we will continue to use our collective strengths to make sure that we achieve our vision of a healthier Nigeria and a healthier world. So on that note, I want to say Merry Christmas to all of us and our families. and ask that we all continue to stay safe as we listen to all the experts this afternoon sharing content and experience with us. Thank you very much, Dr. Anyanwoke.
Thank you.
Thank you, Dr. Okunseri, current ANPA president. Also want to acknowledge immediate past president who is here, Dr. Shemaine Emelife. Our next introduction will be Dr. Adedeji Adefuye, popularly known as Deji. He is one of our panelists for today's dialogue. He's currently the executive medical director of Takeda, I hope I'm saying that right, Pharmaceuticals, and he's an immediate past chair of the ANPA Infectious Disease Subcommittee. He has been very involved in the pharmaceutical industry in a lot of the progressive recent developments, again, pertaining to COVID-19. And he is going to bring that vantage point to today's discussion. Dr. Adefuye, you're welcome. We will introduce the other panelists as well, and then our main speaker will go ahead and give a lecture to the group. After that, then we'll have the panelists participate and lead us in discussions and hopefully have a very interactive session where we can answer all the questions. They invite the tough questions as well. So Dr. Anyanwoke will introduce the second panelist.
Thank you, Dr. Aham. I have the pleasure of introducing Professor Igho Ofotokun. He's a career clinician scientist, and his work, a lot of it has been centered on people living with AIDS. His resume is very long. I will not be able to read it all up because it will take all our time today. But Professor Igho, in a shortened form, is a professor of medicine at Emory University School of Medicine. He's a member of the FDA Antimicrobial Advisory Committee. He's a member of the IDSA governing boards and the HIVMA governing board. He's a member of the NIH Infectious Disease Clinical Research Consortium Leadership Group, which is in charge of coordinating the whole COVID-19 vaccines, not just one, but all of them, and the therapeutics. Without further ado, we welcome you, Professor Igho Ofotokun.
Thank you. And then I'll introduce our speaker.
Thank you. Thank you very much.
Okay, you're welcome. So again, the format is that our speaker, Dr. Obi Nedu, whom I'm about to introduce, will get things going by giving us a lecture on the vaccine. So as a way of introduction, Dr. Obi Nedu is a fellow of the Infectious Disease Society of America. He's an infectious disease expert. He was trained in internal medicine at Tulane University, followed by a fellowship at the University of Washington. He's currently a Center for Tropical Medicine Program Director, Infectious Disease Fellowship at Ochsner Clinic in New Orleans. He's an expert of the Coronavirus Prevention Network and clinical investigator doing research in the COVID-19 vaccines. And so he brings that expertise to the ANPA community today. He's been a thought leader and an authority in his community and also nationally, and has his fingertips on the pulse of everything related to the COVID-19 vaccine. Without further ado, Dr. Obi Nedu, the floor is yours. I believe our technical crew will let you share your screen and then you can go ahead with your lecture.
Absolutely, thank you very much. I am going to share my screen. All right, can everyone see my screen?
Yep.
And everyone can hear. Good. And everyone can hear me okay?
Yes.
All right, so I'm going to go ahead and get started. I was asked to give this talk, and I'm very, very happy to give this talk to everyone. I was also asked to try to be somewhat brief, just so that we can have time for discussion. So if I'm going through slides a little bit quickly, just stop me and ask questions or type in the chat box. I'll try to keep an eye on that. As far as financial disclosures, I don't have any. I just wanted to show this map just to let everyone know kind of the gravity of things and where the hot spots are. I'm in Louisiana and New Orleans. New Orleans was one of the first cities that was hit really hard. Shortly after our annual Mardi Gras parades, we were really slammed. I lost a lot of my patients in the process. And what you can see is that fortunately, Louisiana is not as bad as other parts of the US. This is from the CDC, and I just got this yesterday. I think everyone on this call is familiar with the fact that there's been a number of cases, and unfortunately over 300,000 deaths, some of which are healthcare providers, just like we are. So very somber numbers, and more reason why we need to double down on our public health efforts, and why we're cautiously excited about the EUA approvals for these COVID vaccines. I want to touch base a little bit, just show a rough cartoon of the structure of the SARS-CoV virus, and specifically talking about this spike protein here that allows the virus bind to its target cell, get internalized by the target cell, and ultimately cause some pathology. This spike protein has been the target of essentially all the vaccines, and part of the reason being that it's relatively conserved across different strains of the SARS coronavirus. And the other part being that studies have shown that antibodies to that spike protein are neutralizing, meaning that they prevent the virus from doing what it wants to do. So we're optimistic. We know that people who get infected seem to develop some immunity. There have been very, very few cases of confirmed reinfections. And so most people seem to get some type of immunity. We don't know exactly how long that immunity lasts. As I mentioned, the spike protein is relatively conserved across different strains of the coronavirus. And so what that means is that if you create a vaccine and you create immune responses to that protein, it in theory will still be effective against multiple different strains of the COVID-19 virus. The current vaccine candidates being studied in the US, there are about 6 of them, all showed promise in preclinical non-human primate model studies. And we have phase 1 and 2 trial data on most of them that were generally quite encouraging. We talked a little bit about the fact that the spike protein of the SARS-CoV-2 virus is relatively well conserved, which makes it an excellent target for vaccine development. So stages of vaccine development. One thing that our patients will keep asking us, and you hear this all the time, is, well, this was really rushed. They cut corners. I don't trust President Trump, and things along those sorts of lines. And I think it's important to state that these vaccines have gone through the basic stages of development, the exploratory stage, the preclinical stage. That's when you're testing the vaccine in non-human models, typically primate models, and these vaccines went through that stage. They're currently going through the clinical development stage, which will be your phase 1, phase 2, phase 3 trials. 2 of them have gone through regulatory review by the FDA and have been given emergency use authorizations, and obviously being manufactured and quality control. And so the vaccines have gone through the basic stages of vaccine development. Corners were not cut. Things were just sort of expedited because of the emergency that we're dealing with. So for the clinical development part, as I talked about, your phase 1 trials will typically contain just a small amount of people, and you're mainly just assessing the safety. You want to make sure it's safe in humans. The phase 2, you have a little bit more people, and you're assessing safety as well as immunogenicity. You want to make sure it's safe. You also want to make sure it's stimulating the immune system in the way that you want it to. Then with the phase 3 trials, that's when you're assessing the efficacy as well as also safety as well. And these are usually larger numbers. And so what a lot of the current vaccine candidates for COVID-19 did was they sort of combined phase 1 and 2 and basically assessed safety and immunogenicity with the smaller numbers, and confirmed that those candidates were safe and that they stimulated an appropriate immune response in the individuals that got the vaccines. So again, to be clear, they essentially followed the basic steps of development, and so nothing is no corners are being cut. Let me put it that way. So I just wanted to kind of just quickly talk a little bit about vaccine platforms. The live viruses, we all got measles, mumps, rubella. That's the actual virus that's been sort of weakened or attenuated in a way that stimulates your immune system without getting you sick. Recombinant viral vector, that's when you have an unrelated virus and the gene of interest is inserted in that virus. And that way, when it's injected in you, you produce the proteins from that gene that prime the immune system and get you safe from that particular infection. A good example is the new Shingrix vaccine that's out there. Inactivated viruses will be basically a virus that's killed, and like the flu shot, protein subunits, that's an actual protein from the pathogen. A good example is hepatitis B. You mix the protein with an adjuvant, something that helps kind of stimulate the immune system a little bit extra, and then you inject it in the person, and you end up with a good immune response. And then the mRNA vaccines, these are relatively new, but have been around at least going back to 1990. And what's happened over time is that the technology in terms of storage and transportation of these has developed to where these are now viable vaccine options. So the qualities of an effective vaccine, you really want it to provide long-lasting immunity ideally. You want the vaccine to have really high efficacy. You want it to be safe, limited side effects. The effectiveness needs to definitely outweigh whatever side effects may come from it. And ideally, it should be easy to store and transport. And I'll just add cost effective also. So these are the 6 that have been in different stages of development here in the US. There is a Russian vaccine that apparently is showing relatively good efficacy in preliminary studies. That one is a recombinant viral vector vaccine, and it's certainly shown a lot of promise. But these are the ones that we have under study here in the US, one by Pfizer and Moderna. Both of those are mRNA. Recombinant viral vector vaccines are AstraZeneca and Janssen. And then protein subunit vaccines will be the Novavax, and then the time, I'm not going to go through too much about Sanofi, Novavax. Briefly, the Sanofi is apparently on hold. I think they're still kind of reanalyzing data. Novavax is supposed to be going into phase 3 trials this month, if it hasn't already. It should be going in sometime. Janssen, AstraZeneca, Moderna, and Pfizer, those are all in phase 3 trials already. And we do have some preliminary data from phase 3 trials from AstraZeneca, Moderna, and Pfizer, and I'm going to go through those shortly. So this is the AstraZeneca. This is a viral vector vaccine, and the viral vector here is the adenovirus. And what they did was give a dose of 5 times 10 to the 10 viral particles. These were 2 doses spaced out by 28 days. So this is a phase 1 to 2 trial. So really, they were in this particular trial, they're focusing on on safety, as well as, is it immunogenic? Does it actually prime the immune system? And so it was a 2-dose strategy, 2 doses spaced out by 28 days. The vaccine was found to be relatively safe. And then in terms of immune responses, so this is the placebo, this is the meningitis vaccine. This is people who just got 1 dose. These are folks who got the full 2 doses. And then this arm over here will be convalescent plasma. So this is people who actually were naturally infected, and then you measure the antibody levels a couple of weeks, 2 to 3 weeks after the infection. What you can see is with the prime boost strategy, you're getting antibody levels that are basically on par with those who were naturally infected. So what this meant was, the data from this phase 1/2 trial essentially showed that you're getting good immune responses, comparable immune responses to someone who's naturally infected, and it is in fact relatively safe. So based on this, this particular vaccine candidate went on to phase 3 clinical trials, and these were recently published. And this is preliminary data from this particular trial. And so some of you may have heard of these results already. So at least at the time of publication, this many individuals had been enrolled. And the particular analysis focused on about 11,000 individuals that were enrolled in the United Kingdom and Brazil. And they got the 2 doses spaced out by 28 days. And so I summarized this table a bit on my next slide. I just wanted to kind of say that overall vaccine efficacy was about 70.4%, which is good. If you consider that the flu vaccine in its best flu season would be somewhere between 40 to 50%. And so this is good. But what was interesting about this was there was an error in a small cohort of in this phase 3 trial. Some people got half the dose the first time, and then the standard dose the second time. So LD is low dose, standard dose will be the second time. And for some reason, those who got half the dose the first time and the regular dose the second time, in that subset, the vaccine efficacy was 90%. And then in the subset that got standard doses both times, the vaccine efficacy was lower. And so there's been a lot of discussion about this, and the scientists are certainly looking into that, and specifically looking into how the protocol was altered accidentally, if you will. But overall, the data is very promising for this AstraZeneca vaccine. Just to kind of summarize, those who accidentally got half the dose for the first dose and then the full dose the second time 4 weeks later had a vaccine efficacy of about 90%. Those who got standard dose twice, spaced out by 4 weeks, it was lower. And the overall vaccine efficacy was about 70%. There were 3 cases of transverse myelitis, one that led to a brief halt of the trial while they tried to investigate whether or not it was related to the actual vaccine. To my knowledge, it's not too clear, but it was deemed okay to continue the trial. The other 2 cases were deemed to not be related to the vaccine. And so based on everything, I think it's very promising, this particular vaccine candidate, in terms of its efficacy and safety. And a big plus about it is the issue of storage. It can be kept between 2 to 8 degrees Celsius for up to 6 months, which makes it something very promising, particularly for developing countries that may not have the ability to establish the cold chain for the mRNA vaccines that we'll talk about in a little bit. It also is being priced cheaper than the mRNA vaccines that Pfizer and Moderna have. So a little bit about mRNA vaccines. I think there's a lot of anxiety about it. Again, it's been around for a little while. It's just that we've made advances now in terms of how to store and deliver these. It's not infectious. It generally doesn't replicate in the person. Essentially, it gets into the cell. It uses the cell's machinery to produce the protein of interest. That protein is then presented on the cell's surface, and then it stimulates the immune system. Meantime, the actual mRNA particle is destroyed by the cell. You know, it has a half-life, and it essentially goes away, and you have the protein presented on the surface of the cell, and that sort of is what the immune system sees. It recognizes it as foreign, it builds an immune response, and then that immune response ultimately leads to some memory, hopefully in the long term, in the case of these COVID-19 vaccines. So there are 2 mRNA vaccines in trials here in the US. The Pfizer one, my institution is involved with, and then the Moderna vaccine is the other one. Talk just a little bit about the Pfizer one. I just wanted to show data from its phase 1/2 trials. Again, this is early clinical trials focusing primarily on safety and its ability to prime the immune system. Let's get through that. And so smaller numbers of people enrolled. A few different dosing strategies were used: 10 micrograms day 1 and day 21, 30 micrograms day 1 and day 21, and then 100 micrograms as a single dose. And this is just showing that this is immunogenic in terms of the immune response. So HCS is convalescent sera. So these are people who actually had COVID-19, recovered, and then you measure antibodies afterwards. And what you can see is that vaccine, gives you antibody levels that are comparable, if not higher, than those who were actually naturally infected. And that's really what you want the vaccine to do. The idea of herd immunity from letting the virus run wild and killing people off is, it would be almost unheard of in the infectious disease community. You want to achieve herd immunity through vaccines. And certainly this Pfizer mRNA vaccine appears to at least give a robust immune response and appeared to be safe in these early phase 1 and 2 trials. So based on that, the phase 3 trial went ahead. This was recently published in the New England Journal of Medicine. Again, this is preliminary data. This is data basically acquired 1 week after they got their second dose of vaccine. So still very early, 1 to 2 weeks after they got their second dose of vaccine. So still kind of very short follow-up for these individuals. About 41,000 people had received 2 doses. They used the 30 micrograms concentration and gave doses 3 weeks apart. And they assessed the efficacy and the safety in this phase 3 trial. And I just wanted to point out that there were about 8 cases in the group that actually got the vaccine. That's a little over 18,000 in this group that was analyzed, and about 162 cases in the group that got the placebo. And this led to vaccine efficacy of about 95%. And so we were extremely excited about this in terms of at least its short-term efficacy. These individuals will ultimately be followed up to 2 years to assess long-term efficacy in these individuals. This is just a summary of what I just went over. Again, 95% vaccine efficacy, very, very promising early data. They kind of looked at what they defined as severe COVID, and they were told of 10 cases, 9 were in the placebo group, 1 was in the vaccine group. So the vaccine, again, these are very small numbers, but there's a suggestion that it may protect against severe COVID. And then obviously there's this issue of storage. The Pfizer mRNA vaccine needs to be stored at -70 degrees Celsius. Once it's taken out, it can certainly thaw and stay at room temperature for up to 4 hours prior to use. Just some side effects. The most common was pain at the injection site. That was the most common local side effect, and probably the most common overall side effect that was reported by these individuals. Some of the systemic side effects were headaches, fatigue, muscle pain, but in general, these all resolved within 1 to 2 days after getting the vaccine. I forgot. So that was the Pfizer mRNA vaccine, and I'm just gonna touch on the Moderna. First, going over data from the early phase 1/2 clinical trial. Again, this is also an mRNA vaccine. They used a 2-dose strategy with different concentrations in this early trial. Again, looking to see which would be safe and which would stimulate a good immune response. These injections were given 4 weeks apart, as opposed to 3 weeks apart, which was the case in the Pfizer trial. And I just wanted to show that they did, in fact, stimulate fairly good immune responses. All the different dosing strategies, 25, 100, 250 micrograms. The decision was ultimately made to proceed utilizing the 100 microgram dose with 2 doses spaced out by 4 weeks. You can see this last bar, this is measuring antibody levels. This last bar is convalescent sera. So these are people who are naturally infected, and then you measure antibodies 2 to 3 weeks afterwards. And you can see that your antibody responses are comparable, if not actually higher than those who are naturally infected. And so based on this and the fact that in the phase 1/2 trials, this appeared safe, it went on to phase 3 trials. The phase 3, the early preliminary data from the Moderna phase 3 trial, to my knowledge, hasn't been published in a scientific journal just yet, but it has been released, early data has been released to the press. I just want to say that these preliminary analyses are being generally done by a DSMB. So this is an independent body kind of analyzing the data. So it's not the actual company. I think that's another source of concern that many people have. And in this case, the Moderna vaccine showed a vaccine efficacy of about 94.5% in people who've been followed for at least 1 to 2 weeks after they got their second dose of vaccine. And so again, this is very high vaccine efficacy, very, very promising information. There were 11 severe cases of COVID that were recorded in this cohort, and none of these cases were in the vaccine group. They were all in the group that got placebo. Again, very small numbers, but certainly a suggestion that the Moderna vaccine also may protect against severe COVID. These are some of the side effects that were reported. Infections like headaches. In terms of transportation, this vaccine also needs to be stored at cold temperatures at -20 degrees Celsius. It's a little bit more forgiving because it can remain stable in a standard fridge at 2 to 8 degrees Celsius for up to 30 days. And then it can remain at room temperature after you take it out for up to 12 hours. So this is just sort of a summary. We have our 2 mRNA vaccines that we're excited about because they've shown some efficacy, as well as, certainly the efficacy far outweighs the the side effects that have been reported in a small subset of people in these trials. They've both now been giving emergency use authorization by the FDA. The AstraZeneca vaccine also has very promising early data. Janssen is currently in phase 3 trials, and these other two are a little bit earlier on in their development. I just want to end with myself getting a shot. I was one of the first people at my institution to get vaccinated. And that is my talk. Thank you guys so much.
Thank you so much, Dr. Obinedu. That was very good and very in time. Dr. Aham Onike and I will be looking in the chats to see when people have questions, and we can answer some of them in the chat group. There are others that when we feel the whole group can benefit from it, we'll let the panelists answer them. So please post your questions, and also people can raise their hands for further discussions. Dr. Onike.
Thank you, Gertie. Okay. So Dr. Nedu, thank you for the presentation. Certainly gave a lot of insight into the COVID-19 virus vaccines. As we all are aware, there has been a lot of cynicism and skepticism about the vaccine. As a result, the buy-in seems to be low. Recent studies showed only 70% of Americans are willing to take the virus at this time, which is actually an increase over the last 2 weeks from an earlier number of 60%. So Dr. Nedu, what do you think is the biggest reason for the general skepticism? Some people say the vaccine was developed too quickly. In some other cases, it's the politicization of the process, and then a bunch of conspiracy theories that we see on social media. What's your opinion on this?
I think all the things you mentioned are absolutely correct. I think the quickness, the politics around it, and also the fact that these are new platforms in terms of vaccine development. And I think that that's sort of led to that. And I think it's important that we are able to speak to our communities, Black communities, as well as Black immigrant communities as a whole, and be able to listen. You know, my approach is to listen, to not belittle anyone's reasons for, being hesitant, to listen to them and try to reach them where they are, and try to, offer the best explanation based on what their understanding of things are.
Okay. So we'll go to the chat, and there are a few questions pertaining to your answer just now. Phase 3 trial, how long does that typically last? And does it seem concerning that this was perhaps quicker than usual?
Actually, from that last question, let me just add a few things. And the messenger RNA platform is actually new to vaccines. It is not new. It's been used in development of therapy and certain oncology therapies in the past. So it's not something that is entirely new. It's only that this is actually the first time this has actually been applied to the production of vaccines or to the development of vaccines. So, so when people were actually talking about this was very, very fast, we need to understand that a lot of work had been done previously on the mRNA platform. So the previous work that had been done was the reason why this could actually be fast-tracked the way it was done.
Okay, thank you. So we're getting a flood of questions in the chats. To be able to keep up with all of this, I'll encourage our expert panelists and any others in the broadcast who has an answer to also engage each other through the chats. Myself and Dr. Nyanwoke will pull questions to the general forum as we see fit. It may be difficult to get all the questions answered, so we'll encourage everyone to be as brief and concise as possible and avoid any lengthy preambles. With that in mind, I'll invite, there's a question by Dr. So P. Oshun. P. Oshun, you can unmute your mic and you can ask a question about the number of cases that contracted the virus in spite of having the vaccines. So I'm not sure that went through well. I'll go ahead and ask the question to any of our panelists. There were 8 cases of those on vaccine who got the virus compared to 162 on placebo. And the question is, does that raise any bias? Perhaps Deji can answer that.
One thing I will say is that usually when you recruit for clinical trials, you make a decision in terms of how you're going to randomize those that are actually enrolled in the study. And in this instance, you can randomize 1-to-1, 1-to-2, 1-to-3, depending on really what the endpoints of your study. And then you actually can see that in this study, you had more people actually randomized to what is actually considered the active arm, which is actually the vaccine, and less people to placebo. One thing you need to understand is that it's not every single, even if you go into the real world, and we most likely probably will also change this, because let's also be cognizant of the fact that these studies were done in controlled environments. When we actually start using these vaccines in the real world, the efficacy might be slightly different from what was observed in clinical trials. So it's not every single medicine out there that everybody uses that produces exactly the same effect. That is the reason why it is 95% effective or efficacious, and it's not 100%. If it's 100%, then nobody on the active arm will develop, disease. But because it's 95% effective, that's why some will develop disease. It's important to note that those who develop the disease did not develop the more severe form of the disease as against those who were in the placebo arm. I do not think this actually introduces any bias because I really don't see where the bias here is, because the study was randomized and people were actually randomized based on the randomizing codes that were actually agreed on at the time that the protocol for the studies were written and at the time that people were actually being pulled into the study.
Okay, thank you.
Igho and the OB, because they actually conducted the studies at their sites, they might also want to chime in here.
Yeah, no, I think you answered that perfectly. I was actually just going to comment on a question I saw flash by, if the moderators are okay with that. There was a question that the mRNA vaccines sound like gene therapy. And the question essentially alluded to the fact that this is dangerous and it shouldn't be given to healthy people. And I would want Dr. Deji and Dr. Igho to also kind of chime in on this. But I really think that we should address this, because I think that that's something that is a widespread thought. This isn't gene therapy. So the mRNA is basically just telling the cell which protein to make, and then it goes away. It doesn't stay in the body. It doesn't last in the body. It doesn't insert itself into your genes. It doesn't disrupt your genes. It's like it tells the cell, make this protein, and then it goes. Perhaps Dr. Deji and Dr. Igho, you guys can kind of expand on that. I just wanted to make sure that we speak on that.
If you actually go back into the realms of virology, when you actually look at the SARS-CoV-2 virus, it has 29 main proteins. The spike protein is just one of those 29 proteins. When the genome was actually sequenced from the virus, we're actually able to, really identify the single mRNA that actually encodes the instruction for the development of the spike protein. What essentially happens is, when you receive the mRNA vaccine, what it does is that it actually, because what it does is you have encoded the instruction in this vaccine. So when it gets into the cell, what it does is actually prompt the cell to manufacture spike proteins, to build spike proteins, essentially. That's what it does. It's a messenger RNA, is a messenger, it carries the message to the cell to actually produce these proteins. Immediately, your body actually produces, the spike proteins. Then, your immune system is actually, is alerted. And what happens is that the macrophages actually see this as foreign body, and then the macrophages actually envelope and surround the spike proteins that actually have been produced. That's how the spike proteins, actually will get destroyed, because, the macrophages are going to eat it up anyway. But what happens is that because of the immune response, you have your B cells as a result of this, because your body now says, oh, this is foreign, and the body has to do something about it. Your B cells produce the antibodies, okay, even though the macrophages actually destroy, the spike protein that's been produced. You also have your T cells, your B cells produce the antibody, which actually neutralizes. And then you have your T killer cells that are actually there in terms of being able to remember in the future if you actually come into contact with this virus, and the T cells are going to, and just like Obi said, this cells, what you're giving doesn't last because it is actually destroyed. I don't think it even lasts up to 2 days. What it leaves behind is the memory, which actually is what you have your B cells and your T cells that actually help you. All the stuff that people are actually talking about is gene therapy. It is not incorporated at all into the cell. It doesn't even enter the nucleus of the cell. I think that, we need to understand that. mRNA can never translate into DNA. DNA actually can be used to make it, to send messages for RNA, but not the other way, unless you are actually talking of a reverse transcription. And reverse transcription doesn't happen with SARS-CoV-2.
Thank you, Deji.
So I just wanted, let me just make a quick comment, because this is something that has been of major concern to our community, immigrant community, Black community, and a lot of people that have been concerned about the platform, the messenger RNA vaccine platform. So with gene therapy, we actually alter your gene. You either delete or insert something, and it's almost always a permanent therapy. And I mean, we use it for sickle cell disease and other diseases. In the case of the messenger RNA platform, it's essentially telling the body to do what the virus would normally do if people contact COVID. So when COVID gets into the body, when some people contact COVID, what happens is that the virus is not able to replicate. So it's an RNA virus, and so the protein, the protein making machinery within the body takes the genetic material of the virus, the messenger RNA, and begins to make various components of the viral protein. So what the platform for RNA vaccine has done is to create a way to deliver just a small portion of the RNA of the COVID virus to the spike protein. And so now the body takes that small piece and then makes that spike protein, and the body then develop antibodies to that spike protein. So messenger RNA are very, very unstable. And so within minutes of being delivered, it does its job and is inactivated in the body. It has no permanent effect at all in the body. It has nothing at all to do with gene therapy.
Thank you so much. So we're going to struggle to keep up with all the questions. We continue to...
I think that question was very, very important. I just wanted to add something from a layman's perspective. So think about a cell and think about the nucleus of the cell. So when you inject the vaccine, it goes into the cytoplasm. It doesn't get into the nucleus. The nucleus is where our genetic information is. That's where the DNA is. It doesn't even get to the nucleus. In the cytoplasm, the messenger RNA produces the instruction to produce the spike protein. And that spike protein goes ahead to stimulate the immune system. Messenger RNA self-destructs because it's done with its work. So you have to look at it from the cytoplasm. It doesn't get from inside the cytoplasm. It doesn't get to the nucleus. Therefore, the issue of genetics doesn't even occur. Thank you.
Okay. Thank you. So our three infectious disease experts have obviously made emphatic points about the methodology of mRNA producing the spike protein immunity without impacting the DNA whatsoever. Obviously, it's an issue that has lingered, and I'm glad that we're able to address it, and it may be something that we continue to talk about. However, in the interest of time, considering that we have an overwhelming number of questions, I want to continue to encourage our panelists who are experts in this area to answer as much as they can on the chat. Everybody will be able to see them. And in addition, question by question, we'll pull to the forum. I want to encourage everybody, myself included, as well as our panelists, to be as brief as possible so that we can get through as much as we can quickly. Next question, I guess we can direct this to Dr. Nnedu. From what we know, will this be a lifetime of immunity from the vaccine? If not, how long do we think it will last based on current evidence? And do we think it may be like the flu that becomes a seasonal occurrence as far as people needing a shot?
Yeah, so that's an excellent question. The answer is that we don't know how long the vaccines will be effective. In general, these patients will be followed for up to 2 years, and we'll have at least 2 years of data. And then possibly, and so these are the individuals in the clinical trials, in the phase 3 clinical trials, they'll be followed for up to 2 years. I suspect that periodically from time to time, we'll have data being released, letting us know, how things are going. But the data we have so far is that it appears to protect in the short term. We can't extrapolate too much in terms of long-term efficacy.
Okay, thank you.
Yeah, maybe Igho, maybe you want to talk about the experience from your own sites.
I don't have any different from what Dr. Nnedu has said. I think he said it very well. We do not have long-term efficacy from these vaccines yet. And we will have them because all of the studies that are currently ongoing are designed to last for 2 years. And so we will continue to have information. If you want us to speculate, we can go back to 2002 and 2013 when we had the SARS, the first SARS outbreak in China, and then in 2012 we have the MERS outbreak, which is similar to the SARS-CoV-2. What we observed from those two epidemics was that a number of people who developed antibody to those viruses, the coronaviruses, had long-lasting antibodies. Whether that will be the case with SARS-CoV-2, we don't know yet. And some of the evidence that we're getting from people that have people that have recovered from coronavirus since the beginning of the year, since around November when it was first reported from Wuhan, is that there are several groups of individuals. There are people who develop antibody, significant level of antibody, and their level stays high. There are also groups of people who develop antibody, and over time their antibody to the virus wanes. Whether that is going to be the experience with the vaccination is not known yet, so time will tell.
If I could just add, there also have been, whether the response to a natural infection will be any different from the response in terms of the robustness of the immunological response that actually provides immunity. Whether, natural infection is different from actually taking a vaccine, that is not entirely clear now, but obviously we do know that there have actually been documented cases of individuals who were infected, recovered, and then got infected again. So,
okay. On that line, people are asking if one already had the infection and recovered, do you recommend that they still get vaccinated? Dr. Igho.
I think that is it's a great question. It's something that I've been asking myself lately, because in our hospital, we're having to make the decision on who to vaccinate. And the question does arise, what happens to people that have already had the infection? And I think we have a small bit of data, I think from the FIDSA study, where they actually looked at the serology of people that were vaccinated, who had already had the infection by the time of vaccination. And I think if I remember very well, I don't think it's been published. It was something that was reported in the lay press. My recollection of that data was that there is slight benefit in revaccinating people that have had the infection.
Okay.
Yeah.
Okay, thank you. Next question will be for Dr. Nedu. What percentage of Blacks or people of color were included in the study? Is this a diverse representative study, the Pfizer vaccine?
Yeah, that's a great question. I just want to say that they certainly made a very strong effort to recruit people from minority groups. Now, in the preliminary phase 3 paper, it wasn't specifically reported in the Pfizer paper. For Moderna, I know that initially there was some difficulty recruiting Black and African American trial candidates, but as the study went on, they certainly were able to make that up and were a little bit more successful in bringing on sites that were located in communities that were predominantly Black and African American. Dr. Igho, perhaps you have something to add to that as well.
So we are part of the operational team of the COVID-PM. So we actually are involved behind the scene with all the designs of the study. We track recruitment on a weekly basis for all of the 5 vaccine trials that is being sponsored by the US federal government. When we started out with Moderna, like Obi said, we had significantly low number of minority people of color or underrepresented community represented in those trials. Changes were made, co-VPN sites that were predominantly located in large city where we have Blacks and Latinos were included. So by the time we finished, we completed the Moderna vaccine, 10% of the total recruitment was African-American, about 10%, maybe a little under, approximately 10% were African-American. 15% were Latinos, so which is kind of representative of the population of African Americans as well as Hispanic population in the US. The Moderna trial is also ongoing, and we've been tracking those numbers on a weekly basis. And we are making sure that there are good representation of people from underrepresented communities, and also Black and brown population. So far, I can say, although these are probably not public knowledge yet, that both the AstraZeneca study which is almost near completion. And then the Johnson and Johnson, the Johnson study, which is just recently close to, we've just finished enrolling into that study. The number that is coming out so far are representative. They are kind of similar to what the numbers that we saw with the Moderna vaccine. Yeah. And one thing to note is that we have looked at both the side effect and the efficacy of the virus across age group, across racial group, across gender, and it seems to be, because of the large number of people that are involved in this study, all of them usually minimum is 30,000, we're seeing similar level of efficacy and similar level of toxicity, regardless of race, age, or gender.
Thank you. That's very encouraging that across the board, we're seeing the same results in different demographic groups. So the next question, again, back to the mRNA concern. Are there any chances, from what we're hearing, it doesn't affect the genes themselves, but there's a question about possible carcinogenic effects. Can you refute that? And then also, and we'll leave this for just one of the moderators, perhaps Dr. Adedeji can answer that. And then also out there, are there any other mRNA vaccines? I know the Zika virus vaccine is supposedly mRNA, but are there any others that are fully approved and are in use that may give some public confidence as an example of lack of complications and history of success? So Deji.
Okay, one thing I will say is that, you know, we're going back again to gene therapy. One of the biggest concerns of gene therapy is, you know, either continued expression or overexpression, which is actually why people are always concerned about, you know, the development of, you know, of cancer, you know, in the future. And this is actually different because in this instance, you actually have, you know, there might be gene editing and all these different things that we've talked about that we don't see with the mRNA virus, because Getty actually made it very, very clear. It does not get into the nucleus of the cell. So this risk, you know, developing cancers in the future, you know, is really, it's not there. And you also heard again that this virus, you know, this, doesn't really last that much. It's actually, you know, it's enveloped and eaten up by the viral macrophages in a very short period of time. So that's that. And then secondly, the question about, is there any vaccine that is currently approved? I did mention that earlier. This is actually the first two vaccines approved based on the mRNA platform. I don't know of any other.
Okay, thank you. Next question, in terms of the fear about allergic reactions, how widespread is that? What's the understanding of that? And how does it impact people with known allergic situations, including asthma? Dr. Nedu.
I'm sorry, I was typing a response in the chat box when you were asking me that question. Can you repeat the question, please?
So it was a question from Dr. Osimbowale, and I hope I do it justice. And it pertains to allergy. One of the adverse effects has been an allergic reaction. With that in mind, what is known about that, and how does it affect the recommendation for people with allergic conditions, including asthma, to get the vaccine?
Yeah, that's a great question. I think just to be very clear, these trials include thousands and thousands of people, and there are individuals with atopy and so forth who undoubtedly would be ultimately included in these trials. And so, you know, ultimately when we get all the information, hopefully we'll kind of get some sense of you know, what's specifically what. With the Pfizer, there's been some suggestion that a component in the vaccine, there are people who are allergic to that, a very small number of people, and so they may not get that particular vaccine. And then with Moderna, there's been maybe 2 or 3 instances that made the news of people who had a particular dermatologic procedure, and these individuals were had a reaction. Again, these are very, very small numbers. I think anything that happens kind of makes the news because I think there's a lot of understandable anxiety about things. But I think these are very small numbers, and I think we'll certainly get more information as time goes on.
Okay. Next question will be for Dr. Ofotokun, Dr. Igho. One of the reasons vaccine therapies have been fleeting for some other viruses, including HIV and the influenza virus, is that they mutate. What do we know about mutation with the coronavirus, and how do we think this will impact vaccine therapies pertaining to COVID-19 and other coronaviruses?
This is a great question, and I think it's still a major concern. One thing that we could say, though, is that the target of, so really what makes a vaccine effective across a broad range of strain is to find a target within that family of viruses that is preserved within that virus. And so one thing that we have had, and one luck that we've had so far with the coronavirus, is that there has been report of mutation. And as a matter of fact, before I got into this call, I read from the lay press an article about a different strain of the virus that is spreading in the UK. But the good thing so far is that across coronaviruses, not even just the SARS-CoV-2, all of the vaccine is targeted at the spike protein. So there's what they call the receptor binding domain of the spike protein. That site appeared to be well conserved, and it seems like across the family of coronavirus, it's a well-preserved protein that we could develop antibody to for neutralization. And so time will tell, but I feel like we may be lucky that this vaccine, because of the target and because of the preservation of the target, may be long-lasting and may not be like the flu vaccine where we have to be producing new strains of vaccine every year. But we don't know for sure, but we are hopeful that that may not be the case, that this will last.
Okay. To piggyback off of that, Dr. Igho, does that mean that the flu vaccine in the future may be a target for a similar surface protein that doesn't mutate and then give us a permanent vaccine solution to the flu?
I think that would be somebody who, if we can find a well-conserved protein within the flu virus and target a vaccine to that protein, and so such that it's conserved, and it's that would be a Nobel Prize winning idea. Yeah. So the flu, yeah. So the flu vaccine, the flu virus is completely different, and we've not been able to find a target that is preserved across the different strain of the flu that is that we can develop an effective vaccine for.
But I'm going to chime in a little bit. Make no mistake, immunologists and others are already looking into this technology to use it even to make newer vaccines in future, because this technology seems to be very promising. Thank you.
Good. Thank you. That's very encouraging news. There might be light at the end of the tunnel, not just for COVID, but for numerous other viral concerns.
Yeah. So the other thing I just want to say about the concern about the use of mRNA vaccine. So remember, of the 6 vaccines so far that we are producing, only 2 of them are mRNA vaccines. So it's only the Pfizer and the Moderna. The AstraZeneca vaccine is a vector platform. The Novavax vaccine and the Janssen vaccine are all protein component. So even if you have an issue with mRNA, There are 2, 3, 4 other vaccines that are coming in that will be hopefully sometimes within the next several weeks will have results, and I suspect the result is going to be similar to what we have with Moderna and Pfizer that will be available. So there are other platforms besides messenger RNA that are being developed, and we saw the preliminary result from the AstraZeneca was very encouraging. 70% efficacy overall, it's outstanding.
Awesome. Okay, so next frequent question that comes up is children, pertaining to children. Current recommendations, what's the age range for vaccination? We are obviously trying to get our kids back in schools, and we would want to get them back as safely as possible. But based on current recommendations, what's the age range for administering the vaccine? So Dr. Deji can help with that. Okay, Dr. Nidhi, go ahead.
Well, I think there are no recommendations just yet, at least not to my knowledge. So the ACIP is the organization that would normally meet, and they've been, and they give the CDC recommendations in terms of vaccines. A lot of the trials did not include people below the age of 18. Some are starting to include those individuals now, a little bit later on, and so we'll get some information from that. The early recommendations from the ACIP have focused a little bit more, because of the limited supply, have focused a little bit more on kind of priority groups, which in this case were healthcare workers and elderly people in long-term facilities. As the supply increases, which it will, I suspect that more recommendations will come down the line, and some of these questions might be answered.
Okay, can you quickly touch?
Let me just let me add something.
Just to piggyback off what Dr. Nidhi was saying, can you just quickly touch on pregnancy as well? That's a frequent question that we're seeing. Yeah. And then Dr. Igho and Dr. Deji can make their comments.
I was actually just going to, in my answer, I was actually going to include pregnant women and breastfeeding women.
Okay, go ahead.
It's a standard clinical trial design. Children, pregnant women, lactating women are all, they are all excluded from the study for very obvious reasons. So the first thing that you do is to be able to establish safety and efficacy first in the adult population. And when that is already established, that's when you actually start looking into, the different populations. Normally, in clinical trials, you'll do, what you call a PIP, which is your pediatric investigation plan, which actually builds on the safety and efficacy already established in the adult population before you actually go to children. I think that for the Pfizer vaccine, I think it's 18 and above. And I think for the Moderna vaccine, it's 16 and above. Normally, you do not approve any products, or vaccine for that matter, for a population in which it has not been studied. Because it's not been studied in this population, that's the reason that you're not going to use. It's not approved in those populations. As it pertains to pregnancy, usually when you conduct clinical trials, one of the things you do is to ask those who are potentially could become pregnant, or those that we call, of childbearing potential, to tell them to use, contraceptives during the study. But we do know that in some of these studies, I think Igho and OB are probably better suited to answer that. What I know is just what I've read. There were women, there were women who got pregnant immediately after after they got in the study. And, up to today, as far as I do know, for those two programs, I don't think there have been any reports of, any pregnancy or any other thing. But obviously, like it's done in all other cases, the pregnancies are going to be followed to term. And, and then, I'm actually thinking potentially there might even be a pregnancy registry, in the future, for all these individuals who actually got pregnant while taking the vaccine. So
just let me add to this, because I am part of the operational unit, so I can give you information about the plan for pediatrics and pregnant women. First of all, the Pfizer vaccine included 16 years old and older. So I think it will be safe to say that it could be used in individuals that are 16 years old and older. The Moderna was limited to 18 years old. So all of the major vaccines have now extended. They are now including age 12 to 18. So they are studying now, like Dr. Nadim's, Dr. Obi said. So they are studying now to extend to look at the safety of this vaccine in 12 to 18. As we speak, we're working with Operation Warp Speed to begin trials with the Janssen product, and also Moderna product is in the discussion for children age 5 to 12. And all of these studies are kind of, they are all fast, they are like on fast track. So the whole goal is to get results available so that during the next school season, children will hopefully be vaccinated and can safely return to school. The one thing to do is that now that we know the vaccine is effective in adult population, we don't have to do the large 30,000 people trial in children. All we just need to do is to test whether this vaccine is safe and whether it's immunogenic. It's able to produce the amount of antibodies in children as it did in study. So these are going to be studies that will require maybe 3,000 or less, 600 to 3,000 children, and they can easily be done, and they will be done in months. So stay tuned. So all of those studies are currently in development. And the same goes with pregnancy. So we have within our operational unit, all of this is being run through the IDCRC and the DMID, a study that is a design that is currently in progress to look at immunogenicity of all of the vaccines, starting with Moderna and then extending to the others in pregnant women. So that protocol also is on fast track, and the whole idea is to have this done very rapidly.
Thank you.
Friends, I would like for one of our panelists, any one of you can pick it up, Dr. Adedeji. I have a question here from him saying, I'll read the exact question. The fear of the lack of long-term effect of vaccine is one of the challenges that some patients expressed. How can this issue be addressed with the public based on past experiences and data available?
I think this is more a general question than anything. First of all, you need to really understand all the different types of vaccines, you know, live attenuated and all these different things, some of that actually use, you know, specific segments of proteins, mRNA and all these different things. Even the vaccines that we took when we were much younger, there is a reason why, you know, you've taken all your immunization for tetanus and everything, and then, you know, where you have an injury, you still take the toxoid. You know, there's a reason why you take your, you know, your hepatitis vaccine, and then you actually still go back and all these different things. It's actually based on the robustness of the immunological response, how long it lasts, and, you know, when it doesn't last as long as it's supposed to. You know, the example I always give people is that when you actually receive, you know, the very first dose of the vaccine, it's like just really to tune your body, to tell your body, you know, I really want you to get going and start doing this. When you get the next one, is actually, you know, it turbocharges, you know, the process that you have actually started. It accelerates it and everything. And, you know, how long it lasts actually will vary from vaccine to vaccine. Not all vaccines are exactly the same. So that time those are going to lifetime. There are others that probably will not, you know, Take the flu vaccine, for example. There's a reason why you have to take it every year, because we know that the response is actually not sustained for too long, and that's why you actually have to forget about the mutation and other stuff. The reason you actually take it every year is because the immunological response doesn't last long. And then you have for others like smallpox and all the rest of them and everything, and you take one and and you're done.
Thank you.
Thank you.
Speaking on that, Dr. Adedeji, and perhaps any of the other panelists, what's the understanding between the antibody impact and the T cell impact in terms of providing immunity after a vaccine? So I understand that the T cell, if it's primed, could give lifelong immunity. Is that the case? And how does the current COVID-19, as far as what we understand, have an impact on T cells versus antibodies? Perhaps Dr. Igho can take this one.
So these are all very important questions. These are also very difficult questions that we do not have clear answer for now. And these are some of the things that the people, the immunologists and the virologists who are involved in this protocol are going to be looking into. So for now, we know that when you take this vaccine, it's protection. So the question you're really asking is, what are the correlate? What component of the vaccine? So there are two components, the cellular response and the antibody response. Which of these is the correlate of protection? And I'm sure among those 162 people who had infection in the Pfizer and the 8 who had in the Pfizer control arm and the 8 who had infection in the vaccinated arm, we're going to be taking a look, dissecting all of those data, and we're going to be looking at what is really providing the protection. Is it the cell response or is it the immunological response? We do not know for certain, but we do know that from the phase 1 study and some of the early basic immunologic studies that have been done, that these vaccines generate both robust antibody response, and they also generate very robust immunologic T cell response. But stay tuned. And then with regard to the previous question that was asked about the duration, what are the guarantee that this may be a long-lasting, will provide long-lasting immunity? Again, like we said before, we don't know. But there are a few things, previous data that we can speculate. For example, we know people that develop natural infection. They, even though there are cases of reinfection, they are extremely, extremely rare. And so based on that, people think that natural infection may provide some form of protection. And some of the people that have reinfection, they don't really get a severe infection as people who are getting their first infection. And then the second was what I mentioned earlier on, the SARS COVID infection that occurred in 2002, and the MERS infection, the MERS epidemic that occurred in 2013. We know from those two epidemics that people actually develop some durable antibody response to the coronavirus. So time will tell, but we are cautiously optimistic that this may provide some reasonable level of protection, but nobody can say for certain.
Along those same lines, we'll bring in Dr. Nnedu. Most of the assessments as to potential diminishing immune capability with time has to do with measuring antibody levels in the serum and seeing that they decline over time. If it is indeed true, as Dr. Igho had suggested, that there is a cellular response with T cells, do we have a way of measuring it, and what do we think in terms of, again, a hope for lifetime immunity?
Yeah, very good question. So there are sort of proxy ways to measure cellular immune responses or T cell immune responses, and certainly those are those are being done with these trials, and more data will certainly come up along those lines. And so I think, as Dr. Igho said, more information will come. I will say that from phase 1/2 trials, it does seem like they stimulate T cell responses. But I think we'll get a little bit more information as things come along.
Okay, thank you. So it's great to see all the questions being answered by not just our panelists, but other experts in the group on the call today. And it just shows the expertise that we all have in the Nigerian community. With that in mind, what about Nigeria? What about the Nigerian population? Amongst the vaccine that have crossed the finish line, Pfizer and very soon Moderna, and then some others, including those from China, Russia, and Europe. What's the recommendation? What you project or assume will be the solution for bringing the pandemic to an end in Nigeria? I'll let all three, actually all four, including Dr. Anyawoke, make some brief, hopefully brief comments on that. Beginning with Dr. Nedu.
Yeah, that's a very tough question. Yeah, I think I alluded to some components of a good vaccine in terms of ability, storage and transportation. And so I think that whichever one is chosen for Nigeria will be one that would have relatively, because vaccines are already administered in Nigeria. And so it will be able to just use, existing storage and transportation, channels. And so the mRNA vaccines obviously require, significant cold chain that's more than sort of the other standard vaccines. And so, my suspicion or my hope is that, the AstraZeneca and the Janssen vaccines, which don't, those vaccines can be transported and stored in kind of more traditional mechanisms that exist in Nigeria. And, if those are effective, and it's looking like those will probably be cheaper than the mRNA vaccines, my suspicion is that those are the ones that will, get disseminated back home.
Okay. So the rich Nigerians will get the mRNA and the poor Nigerians will get.
So just to add to that question about what the government of Nigeria is doing, by the way, I agree with what Dr. Nedu said. I think on December 2nd, because we've been working, ANPA has been working with the Nigeria Federal Ministry of Health, the COVID and the Nigeria Governors Forum. So I reached out to the director of the Nigerian Governors Forum in terms of the medical director and asked if the Nigerian government has put any plans in place for Nigerians. And his reply to me was that they have an expert panel, expert review committee that had been constituted by the Minister of Health, and about 44 million vulnerable persons have been estimated to require the vaccine at a cost of about $92 million. So that's where Nigeria is as at this time.
I just wanted to say that I had, I was on a call before this particular call with some high government officials, and the rumor I heard was that they expect, the federal government is saying that they expect to have vaccine in Nigeria in January. And I also just wanted to make sure that none of these vaccine is necessarily superior to the other. All of them have the same, it's just antibody to the spike protein. And regardless of how the spike protein is delivered to the body, the response from what we're seeing from all these studies, that it's similar. So I expect that the efficacy and perhaps some of the side effect is going to be comparable across all of the vaccines that are being developed for COVID. So regardless of what product is available in Nigeria, I think it will be fine. And I think I saw a question about on the chat about whether the Pfizer or the Moderna, which is preferable? My answer is that go for the one that is available to you. I am scheduled to have my vaccination today after this meeting, and I'm going for the Pfizer. I'm going to be receiving the Pfizer vaccine. We developed the Moderna vaccine, the first dose during the phase 1 study. The first person ever to get the Moderna vaccine was here at Emory. It was my colleague, our team, that started the phase 1 trial, and we're very familiar with that product. But I think all of them are going to be comparable in terms of efficacy, and to a large extent in terms of reaction or side effects.
If I can just add, just add this little wrinkle. Many of us speak to people in Nigeria and all these things, and then of course, we actually led the discussion with the federal government and everything. I will say, whatever it is that they're saying, the bottom line is that we don't have any good plan. And the reason I say we don't have any good plan is that from my discussions, including people inside Nigerian CDC, is that the federal government has not even contacted or contracted any of these companies for the purchase of vaccines. Now they're talking about, you're looking at the, population that you want to vaccinate without procurement. I think procurement comes first. You do procurement, and then you actually go into who are the folks that when we do get the vaccine, then when is it that, you actually will be, who are the folks that you actually will be able to vaccinate. So that is that, where it's wait and see for us. But for me, as a person, I don't see any structure in terms of what we really want to do. I have not seen any concerted government effort that indicates that the government is actually taking this as serious as it should. So, and since I'm speaking, I want to actually quickly use this to address some questions that people were asking about convalescent plasma. And convalescent plasma is, when you actually use convalescent plasma, you are essentially using plasma that contains all kind of different antibodies together and everything. My company, along with a few other companies, actually in a consortium with NIAID, and we're conducting a study that actually focuses on extracting just the antibody to SARS-CoV-2 alone. And maybe some of you have heard of it. They call it hyperimmune globulin. And the reason I'm actually mentioning this is that I was actually able to push NIAID to actually have a site in Nigeria. The National Institute of Virology in Jos is actually recruiting as part of that study. This is something that's not happened in Nigeria, for a very long time. And Nigeria, you'll be surprised, actually right now has recruited more people into that study more than UK. So that's actually something good to do, to know right now about Nigeria. But in terms of the vaccine, I really don't see much.
Okay, we're going to Dr. Charmaine Emelifede, the current past immediate president of ANPA, has her hands up. So we're going to give Madam President the opportunity to ask her question or say what she has to say.
Hello, can you hear me?
Yes, ma'am.
Yeah.
You're echoing. We really can't hear you. I think you're probably around.
Dr. Emelifede, we can't hear you.
We don't understand.
Yeah, I think we'll have to come back to her.
We
can't hear you, Charmaine.
For the last 1.5 hours, and it's an awesome conversation.
Madam President, you will have to ask your question.
Can you hear me now?
Yeah, we can hear you now.
Okay. I was just saying thank you so much. We've been listening for the last 1.5 hours. Can you hear me?
Yes,
yeah,
ma'am.
we can hear you better now.
Okay, fantastic. I think this is a solid team that we have speaking, and the beauty of this is that the people listening to this are not just physicians from ANPA. We also have the general community listening to this, and so it's a testament to the great work that ANPA is doing in helping to educate the community about this very important pandemic, and obviously the vaccine that's in front of us. Of course, little did we know a few months ago when this started in February, March, that we will at this point still be talking about it, more so have a vaccine. And this COVID-19 task force and you guys as the experts have really continued to educate the membership. So we really, really do thank you. Hopefully, Dr.
We lost you again, Dr.
Yeah,
Emele.
we lost her. Okay, we lost her.
What
Letitia, we may need your help to
doing will trickle down to the Nigerian leadership. They can learn from what is being said. All right, go ahead. Go ahead.
Thank you. Thank you, Madam President. Dr. Hezi Adesanya.
Hi guys, very informative sessions. Sorry I came in late, rushing in from another meeting. Just for general informational purposes, there is an ongoing discussion at WHO level with GAVI, that's General Alliance for Vaccines and Immunizations. I just got that information yesterday. One of our Nigerian scientists is on that board negotiating bulk pricing at heavy discounts for third world countries, specifically Nigeria. So I guess over next week, we'll know more about that story publicly. My question has to do with safety, and I'll direct that to both Dr. Adedeji and Professor Igho, Ofotokun. What is the difference in terms of immune reactions with the Pfizer Moderna vaccine, which is mRNA, versus the AZ vaccine, which is attenuated virus? What do we have we seen anything, or is there anything different from a safety perspective that we expect, especially dealing with vaccines based on the makeup of the two products? Thank you.
So if I may go first. So when we think about, so first of all, comparing the Pfizer Moderna mRNA platform with the AstraZeneca platform. Let's talk about the mRNA platform. So what the vaccine contains, there are two components. You have the mRNA itself, and then it's packaged with what they call a lipid platform. And so you deliver the mRNA and the lipid platform. And so the reaction is either to the messenger RNA that is being delivered or to the emollient or the platform by which the virus is being delivered. And so it looks like with mRNA, people, especially people with allergic reaction or autoimmune disease, tend to have more reaction to the mRNA component of the vaccine. And then, of course, in general, people have a reaction to the lipid component. And from the trials that we've done so far, most of the reactions are really either local reaction, people have bump and pain at the site of injection, or people have this general, what they call reactogenicity, that we see with almost every vaccine. So fever, and then people have the malaise, headache, all of those last usually a day or two and it resolves. And then in a very small proportion of individuals, you have this so-called allergic reaction. And it seems like this seems to be more common in people that tends to have allergic reaction, that have autoimmune disorder. And so those are the type of people we're recommending. If you have previous heavy reaction, if you have autoimmune disorder because of the mRNA component, you're likely to be susceptible to those type of reaction. But thankfully, they are extremely, extremely very rare. And what we're seeing with the AstraZeneca product is not different. We're having the same type of reactogenicity, mostly local reaction. And then again, we have a lot of these malaise, headache, and all of them tend to go away. It actually seems from, we don't have the full data yet, it looks like from the people who are doing the study, they think that some of them really, if you take like any of these pain medication, like Motrin, for example, Tylenol before you're vaccinated, a lot of those effects are kind of mitigated. It's not as bad. I don't think that there's a really huge difference yet in terms of the reaction that people have to this vaccine. I think they are comparable across the board. And when you compare it to other vaccines that are out there, so you compare it to the flu vaccine or you compare it to the Shingrix, which is actually the one that has most side effect of most of the common vaccine, we are kind of in between the flu and the Shingrix vaccine for shingle. So what we're saying is not out of ordinary for what you expect to see for vaccination.
Yes, I probably wouldn't have been able to say it any better than Igho did. So what I'm going to say is actually going to be limited. One of the things that we need to understand is that the body, when you're talking about, allergic hypersensitivity reactions and the like, it's usually when you actually have, foreign proteins actually, injected into the body, or your foreign proteins actually. And, these vaccines are not actually, they are not foreign proteins. These are actually, the mRNA, for example, actually is, it's just supposed to actually ask the body to produce its own protein. So that's why, hypersensitivity and allergy are actually also, reduced. The other one is actually the AstraZeneca is actually a viral vector. And we know that viral vectors have actually been used, as a transporting agent for very many different, medications and, and vaccines. And, except for those, as Igho said, who actually already have, immunogenic, liability for them to actually have all these allergic reactions. I haven't seen much, and, it's wait and see. But based on, the way the vaccines are produced, the mechanism of action and everything, I don't expect to see much except those local and maybe a few systemic things like headache and all the rest of it, which actually have been extremely, extremely rare based on data coming out of the studies.
Okay, sorry, quickly, one very short minor follow-up question to you, Deji, and that's because you're in pharma, so you might, Igho himself is closely into this. Stability question. We're dealing with vaccines. We all know Pfizer minus 70 degrees, Moderna minus 24, close to normal fridge temperature. Again, my question is trying to distinguish between the mRNA platform and the attenuated virus. Do we have what do we expect? Because we don't have real world data yet. In terms of stability, do we expect any differences with this product when has it been tested? Do we know data about it or whatnot, whatever? Because now that it's out in real world, batches will be withdrawn and tested. And do we expect any difference between the mRNA platform and the attenuated virus? Sorry, I keep going back trying to differentiate that.
The answer is, I don't know.
Yeah.
One thing I can tell you, one thing I can tell you for a fact is that when these vaccines are transported, when the Pfizer vaccine was getting close to that point, they actually provided, you know, containers that you actually can use to transport the vaccine. And of course, we all know about, you know, how we actually preserve organs that are harvested before they are transplanted, you know, with dry ice and all these different things that you have to do, you know. Also, all those are in place, and Pfizer is providing, along with the vaccines, all these containers to be able to transport them. And the other part of it is that each one, you know, every time, whether it's vaccines or any other drug for that matter, when you put them into the market, you always have, you know, you always have samples that you hold back for testing. And that's how you are able to actually make a determination of how long they're going to last on the shelf. When you get them outside, how long they're going to last. And all these are being continuously monitored.
Yeah. Just to address, just to add to what Deji said, so the stability is really the biggest disadvantage of the mRNA platform. So mRNA is very, very unstable. I mean, it degrades within minutes. And so that is why they have to be stored in very, very cold condition. So for the Pfizer, it requires -70-degree temperature. For the Moderna, -20-degree. But for the AstraZeneca, which is a vector subunit, and the other vaccine, Novavax and Janssen, that are being produced, they are a protein subunit, will not require that level of, they are more stable and will not require that level of cold to preserve them. And so that is why people, and I think Dr. Obi pointed this out in his presentation, you can keep the AstraZeneca vaccine in at just regular refrigeration temperature, the regular fridge, for as long as 6 months, and it's stable. But you couldn't do that with either the Pfizer or the Moderna. And that is why it's probably, the people are saying the AstraZeneca is probably going to be what many people in resource-limited setting will be able to use. But in terms of efficacy, I think they are comparable.
Okay, to get back on what Professor Igho just said, if you remember when Dr. Nedu was presenting, he showed that it was the mRNA had to be put in a lipid molecule, which is what is called the nanoparticles. Yeah. So they needed that to preserve the messenger RNA. And Dr. Deji keeps saying it will destroy easily. So in that lipid molecule, it gets injected into the muscle. And that lipid molecule, I guess with the temperature of the body, it fuses with the cell, and that opens it up to release the messenger RNA. So I think that difference in the temperature is more to do with that lipid molecule. And the way that Pfizer, BioNTech did their own lipid molecule is slightly different from the way that Moderna did theirs, hence the different temperatures. Thank you.
Yeah.
Thank you. So with all of that in mind, again, back to Nigeria, you know, the evidence so far seems to indicate that the impact of COVID-19, in spite of poor preparation, poor infrastructure, and arguably questionable leadership, has still resulted in an impact of the virus that appears on the surface to be less than what we're seeing in Europe and North America. With that in mind, as far as risk-benefit goes, does Nigeria really need to invest heavily, $92 million perhaps, to vaccinate a handful of people? Is that a worthwhile investment in comparison to the current understanding of the impact of the virus? Dr. Nedu and then Dr. Anyanwoke.
Thanks. That's a very good question and a tough question to answer. I think from my perspective, I think it's a little tough to say just how the epidemic is in Nigeria. I think certainly perhaps we're not seeing the excess deaths over the usual number of deaths that one would see. And that may suggest that we're at least not seeing as much mortality. But in terms of the transmission in Nigeria, I'm not sure we fully understand that. And I'm not sure we fully understand why we may not be seeing as much excess death compared to previous years. And so, you know, my father still lives back in Ofotokun. He's 80-something years old. we're not too sure. But you know, I certainly would, you know, I think that it's important that the Nigerian government make it a priority and make the investment in terms of vaccines. And hopefully there'll be a global, I think someone alluded to the fact that the WHO is working to make some type of global push so that these vaccines are available everywhere.
Okay, to chime in.
Go ahead, I think that's your question.
Okay, to chime in, I believe that there are some work still ongoing as to why we're witnessing the phenomenon we're witnessing in Nigeria in terms of us not having it as much as it is over here. If you compare the Africans and the people in this climate, it doesn't seem to be genetic, the virus itself. So in my opinion, Black people are more likely to have comorbidities like hypertension, diabetes, obesity, and they may not be managed optimally. A lot of times back home, some people will go to the doctor, they tell you have hypertension, give you medication for 1 month, and they think that the hypertension is cured. And so they stop taking the medication. So you can see why a lot of people will be susceptible that are Black. People who live in densely populated areas and overcrowded living arrangements may predispose to more COVID infections. A lot of Black people, especially minorities, seem to do more of the direct service jobs that leads to predisposition. Now, this is in America. And then there's access, the issue of access of care, which if you go back to Nigeria, there's still the issue of access of care. It's a lot that is going in Nigeria that we don't really know. But I think that I want to say that the climate, the tropical climate might be helping for it not to be as much as it is here. But I hear from my friends that are in Nigeria that the numbers are beginning to tick up again, even in Nigeria. So a lot of this stuff, it is pertinent to know that why we may be noticing the same rate of infections and mortalities in Nigeria, we still need to figure out the exact reasons why this is the case. People already, there was this particular study, it wasn't particularly a very good one, that said something about the BCG vaccination. We don't know much. And in Nigeria, it's not a society where they jump on research. Let's figure out why this is so here and why it's not so here. But in a nutshell, like I said before, the Nigerian authorities seem to think that they have about 44 million people that are at risk and that they will be planning to vaccinate. I guess we'll wait for early next year and see. I hope that answered the question.
Yeah. You know, one thing I probably will add is that, you know, all this issue about BCG vaccination, use of anti-malarials and all those things have been debunked based on, you know, if you take the Indian population, for example, they kind of mirror, you know, the environment in Nigeria to some certain extent. And that was, it was in India that they actually looked at all these things about vaccination and all those things and actually found out that they really do not really truly reflect. You know, one thing that is actually really for, you know, I work for industry now, I don't do studies anymore. For those of you, you know, B and Igho, you know, I've actually discussed, you know, with a colleague at Lutz, if they actually are able to get the kits to be able to do, you know, assessment of SARS-CoV-2 antibody, for them to just do, you know, a surveillance of some sort and randomly maybe grab, you know, every other person that comes into the hospital and test their antibodies. I'm sure that the numbers that we're seeing from, you know, it's more like a sentinel, you do like a sentinel study, you know, the numbers that we're seeing in Nigeria, you will know that it's not even close to. I'm sure a lot more people in Nigeria get infected that they don't know, and then, you know, just don't feel anything. So all the numbers that we're actually getting, I'm telling you, those numbers are not reliable because I think a lot more people are getting infected. But why, the reason why, even if they're getting infected, that they don't exhibit, you know, the more severe disease is still something that still befuddles, you know, most of us. So Obia and Igho, You guys still have the capacity to write grants and everything. I stopped doing that a long time ago. So something for you guys to take on.
Okay, thank you, Dr. Deji. Yes, I think that's the take-home point, is that we all realize that the numbers in Nigeria are underreported, but the mortality, the number of people you're seeing with respiratory symptoms and ultimately dying seems to be far short of what was predicted. And there's an explanation for that eludes us for now. And that explanation could help determine the best public policy. But I guess with time, hopefully we'll have some answers. So this was supposed to wrap up at 4. Obviously, the appetite remains strong, and the panelists seem to be willing to keep going. We can give it an extra 10 to 15 minutes, take a few more questions, and then have final comments. I see Ms. Lucy Obasuyi has a question. Lucy, you can go ahead and unmute yourself and ask your question to any of the panelists.
Thank you so much. I want to present this to Dr. Igho Ofotokun. I'm sorry if I'm pronouncing that wrong.
Ofotokun.
Okay, Ofotokun. Okay, sorry. Thank you. My question is: everybody understands that this vaccine is present, and there are so many misinformations about it. Okay. This is a medical, from what you are explaining to me, it's like I'm in the medical community, but I'm not. So I was hoping that this is what we need, but how do we get this, translate this to ordinary people like us who are not in the medical community, understanding that once you get the vaccine, what happens? Am I going to stay after I go through the phases? What do I do next? I still will be wearing my PPE, or I'm not wearing that anymore. Or these are the questions that are puzzling to a lot of people. I think the misinformation, this is giving us the opportunity to enlighten others. Like I can carry this to others about how important this is, because a lot of things I'm seeing about this being some kind of gene manipulation. I mean, we're reading that everywhere. But we need to understand why we need to take it. Why do we need to take it if we have to still be using PPE after we take it? What is the duration of time that this will, when we can stop wearing the PPE? Something like that. We need to understand that in a layman's language. Thank you so much for everything you guys are doing.
Lucy, before you sign off, how do you find out about this meeting, and what organization are you part of? And that will help us see how we can reach you, your colleagues, and the larger community. Just give us a piece of that information, please.
Yes, please. I'm a member of the United Nigerian Women Organization, and Dr. Kidi is our president. That's how I got the information to join the Zoom.
Okay.
Thank you.
It's a challenge to us to figure out how we can reach the rest of our community. And I think many of us are open to an invitation where we can have discussions like this and allow us to have that kind of granular penetration. So thank you for joining us today and for your questions.
Thank you so much for having me.
Thank you. So Dr. Anyawoke, do you want to take on her question?
Okay, I thought her question was for Dr. Ofotokun.
I'm sorry, you're right. You're right.
I can take it though, but I will let Dr. Ofotokun do it.
I think Dr. Anyawoke will do a good job. But thank you very much, Lucy, for joining us, and thank you for that great question. I think so we have to, it's our frame of mind. We have to think about this as we are all in it together. We're not thinking about just me or my family. We're thinking about the entire, this is a pandemic, the entire world is in this together. So my action has two major aims. What do I do to protect myself? What do I do to protect my neighbor? How can we all work together to protect each other? And I think if you approach it that way, then it becomes easier for some of the public health measures that are being recommended, becomes easier to adhere to. So I'm a physician. I have to wear masks 24/7 in the hospital. I can tell you that it's not fun. I don't want to. But I wear it because I want to protect myself, but also I want to protect my patients. And so we do it for one another. And so with regards to your specific question about after vaccination, when would all of this be over? This will begin to ease when a significant number of us have been vaccinated and we are protected against the infection. So what that means in medical terms is that when a large number of us have achieved herd immunity. So when there's herd immunity, usually that takes about 70% of the population. So if 70% of us have been vaccinated, it becomes difficult for the infection to spread within the community. And I think until then, even after vaccination, which I'm going to have my vaccine today, I'm still going to adhere to all of the public health measures. I'm still going to wear my mask, keep my social distance to make sure that I do not, remember the vaccine is not 100% protective. It's 95% protective, but there's still a small chance. If I get it, even if I don't get very sick, my next door neighbor who is 87 years old or my patient who is immunocompromised may get it and may lose their life just because of their contact with us. Yeah, but I can see that by the end of, I'm thinking sometime towards the end of next year, all of this is going to ease down and life is going to begin to return to normal. Thank you.
Thank you so much for that. Appreciate it.
Okay, we have Dr. Odira Nwankwo's hand up.
Thank you so much, Gary. I think you guys have been doing a great job. I know that so many people are still very skeptical about taking this vaccine, and probably rightly so. From the discussions and from what we know, the mRNA platform is new. There is no vaccine out there that has used this platform. I wanted to find out from the expert panel, do we have any other biologicals, any other therapeutics out there that has used the messenger RNA platform, and what has been their safety profile? I would think that if we have such products out there and those are framed into how this is communicated to the populace, that might begin to allay some fears that folks might have.
Okay, I'll just give you, my industry perspective. I did mention earlier that, this is actually not an entirely new platform. It only just meets something that had just been used to manufacture vaccines. It's been used for gene therapies before. And I think there are at least, I can't remember the programs because I was actually checking these out. There are actually programs that I think one might actually be in phase 3 development for gene therapy that actually uses this platform, and the safety of the safety review concerning that program seems to point to a lot of the things that we've actually talked about today, that the risk associated with using the mRNA platform as against using a viral vector that is used for gene therapy in a lot of cases in which you have off-target overexpression, prolonged exposures and all these things. You actually don't see them with the messenger RNA platform. And this is really what gives, because we talk about all these different things. This is science. It's really going into the pathophysiology, the mechanism of action. How does this thing actually work? So when understand how it works, there are no off-target effects and everything. Very labile, a virus platform that actually disintegrates very quickly, and the fact that the macrophages actually do their work in actually mopping up, excess spike proteins that are produced as a result of, the encoded protein that actually encourages the development, the encoded MES instructions that actually encourages the development of these spike proteins. They are all mopped up. So that is really what, when you look at all of that, this is actually what gives people confidence that, no off-target effects, and the fact that, you know, it gets destroyed over time, that they know long-term, there won't be any long-term, you know, any long-term effects. But, you know, we'll wait and see. You know, like studies are done under controlled environment. When we get into the general population, we'll see. But I personally don't see anything, you know. My daughter got her own vaccine yesterday. She asked me all those questions because she works in the hospital. Told her to go ahead. I'm getting mine next month. So, you know, if I have concerns about it, I won't be asking my daughter, because the main thing she was concerned about is like, okay, my daughter just got married. She's like, they say this thing is going to make me infertile. I'm not taking it. I say it's all, you know, all lies. Don't listen to any of these things. You're going to be protected. Just go ahead and get it. So she actually took it yesterday and she sent me a picture. So, and I'm getting it too. Everyone in my household is going to get one.
Yeah, it's excellent points, Dr. Deji. You know, I think we're in an era where the cynicism is at an all-time high. And, you know, I'm an orthopedic surgeon. I'm not an immunologist, but myself studying the methodology of the vaccine, it's actually, you know, groundbreaking and extremely impressive, and it should be celebrated. But instead, we have the opposite reaction to a large extent. Personally, remember being in school in Owerri, and at some point they called all of us out and assembled us in line and shot vaccines into our shoulder, one student at a time. And I got home and my mom was livid because the school had somehow permitted some agency to do that without consenting the parents. And she was worried that there were cases of polio, that some people got real polio after those kind of vaccines. And so we're not even talking about those kind of complications with the coronavirus vaccine. We're talking about allergic reactions, and yet the public skepticism is unbelievably high. So it's obvious that as a medical community, we're failing to connect to really fully assuage those concerns. And you know, the onus is on us. Your own daughter getting the vaccine, obviously, from your recommendation, speaks for itself. I think we're almost out of time at this point.
Yeah, I think Igho was going to say something. Maybe we should allow him to say it
yes,
to close it for us.
I was going to yield to Dr. Anyanwoke, but yes, the panelists are welcome to make some parting remarks. Of course, any of the ANPA executives, including Dr. Kunseri, if there are any comments to make, Dr. Edionwe and the rest of the people who put this together. Dr. Anyanwoke, it's your call. We can allow a few more questions before we go to closing remarks.
There's a young lady's hand that I've seen up for a while, and I feel bad if we don't call her. Her name is Melissa. So Melissa, please be brief. Thank you.
Yes, hello, thank you. I want to thank all the panelists. I'm actually a student and I've gotten a lot of information for everything I've heard today. But my question was, I know there's a lot of people who are taking things like zinc or garlic tablets, other herbal supplements. I was just wondering if there's any contraindications or is there any synergism with those kinds of medications with the vaccination? And on the opposite end, if there's anything that's been shown to help boost the immunity vaccine.
If you can't afford your medicine, thank you. Well, it's open to any of our panelists.
Thank you. That's an excellent question. I think there's been a lot of discussion about those things. I just want to say that there's no clear data that shows that I've heard about zinc, I've heard about vitamin C, I've heard about different vitamins. There's no clear data that in any way leads to greater protection or that it alters the course of disease if someone has COVID-19. I usually just advise people to have a healthy diet that provides them with the necessary supplements, with the necessary vitamins and so forth. But there's, I guess that to make it short, sorry, is that there's no data that it helps, but it probably doesn't harm you to take that.
Okay, Ms. Melissa, I hope that was okay with you. I would say that I do advise people to keep their immune system healthy, and vitamin C, vitamin D, zinc all help with our immune system. There's nothing to prove that they help us to cure coronavirus. So if you're talking about immune system, that's fine. And there's no data to show that it interacts with the vaccine, if that's your question, okay?
Great, thank you.
So we are at 4:20 p.m. Eastern Standard Time. We've gone 20 minutes above our time. I hope you guys know that we have another of these next week, Saturday, same time, 2 to 4 p.m. We see that the interest is strong, and thank God we have this other one scheduled for Saturday. Please, please join us at that conference. Bring your questions with you. I will keep answering it in the chat, and our panelists will keep answering them as we go. I am so sorry, but at this point, we have to wind down. I'm sorry, I see all the hands that are raised. I am sorry. We'll do this again next week, Saturday, December 26th, 2 p.m. to 4 p.m. At this point, I'm going to call on our ANPA IT chair in the presence of Dr. Mercy Ejionwe. You kind of help us to wind down.
Okay, thank you, Dr. Anyuoke. This is Dr. Mercy Ejionwe. Hi, everyone. What a great discussion that we had. What great information that we learned. I hope everybody got a lot of their questions answered, and I hope you guys are available for the next session that we do have. I just wanted to take a time just to say, for all of those who want to know more about our different events that we have, just like this vaccine conference and other events that ANPA conducts, please follow us. And you can follow us either by going to our website, www.anpa.org. We're also available on our social media platforms and the Facebook, the Twitter, as well as Instagram. And I actually put our flyer on the actual, what do you call it, the chat, and I'll also write it down as well. And so if you guys have any questions, let us know. Again, thank you guys so much for joining us, and hope to see you guys again at the next one. Thank you, Dr. Gertie.
Thank you, everybody. Dr.
Thank you.
Oh, wait a minute. We still have Dr. Dr. Odira Wankwa to give us a vote. Thank you. Dr. Odira.
Thank you so much. I honestly do think that this has been a very robust and engaging and interesting discussion tonight. On behalf of the ANPA COVID Task Force, I want to especially thank the expert panelists. I learned a lot. You guys did amazing. Dr. Igho Ofotokun, Dr. Obi Nedu, and Deji. We learned a lot from you guys. I especially also want to thank the ANPA National Executive Team. I want to thank Professor Chris Okunseri. I want to thank Shaman, Dr. Shaman Amelife. I don't know if Chinyere Anyog is also attending. If she is, I also want to thank her. I especially want to thank Letitia and Britt for working at the background to be sure that we'll have a successful discussion that we had today. I will not finish this without thanking the moderating team, Dr. Getty Anyuoke. You guys put together a very nice discussion. Ahamo Onyike, you did a great work. I also want to thank, I think it's Mercy Ejionwe that just spoke from the IT, and Susan Enyonwe, I think she's the director of ANPA membership. Thank you guys, I love this, and I will sincerely ask that we do this again. Thank you, Merry Christmas, Happy Holidays, and prosperous New Year. Thank you.
Thank you.
Thank you, thank you.
Thank you. Bye, everybody.
Bye.
Okay.
Bye.
Have a great weekend.
Bye.
Bye.
Thank you so much.
Thank you. Thank you so much.
Yeah, truly, thank you.
Thank you so much.
Who's that?
Imela.
Imela, who's that?
Abumo.
Oh.
Hey, how come some that Malia since some come judge?
I'm so sorry. Can you join us next week, please?
No, that was great. Good.
Thank you, sir.
That was good.
All right.
Thank you so much, Doc. That was great.
Thank you, sis. Thank you so much for logging in. We are going to do this again next week, same time.
Yes, ma'am. Bye-bye.
Bye.
Thank you so much. That was a nice one. God bless you all.
Thanks, sis. Thank you. Please show up again next week, Saturday at the same time, 2 to 4 p.m. Eastern.
All right. Thank you. Bye-bye.
Bye. All right.